Many of you have had the experience of being graduate students, and at various times during your stay as a graduate student, you have an examination, and sometimes you fail the examination, in which case you're allowed to come back in a few days or a few weeks. So I'm back. You can take that as you like. Let me just make a few remarks about this group that I'm going to talk about. I'm not going to, unlike much of what you've heard, tell you, good, hard scientific facts. There will be some good, hard scientific facts because I want to convince you that I'm a scientist. But a lot of this is speculation. And the interesting thing about this subject, or one of the interesting things to me, is A, it's a really, really interesting area. Where does life come from and what is it? The second thing is that there is zero agreement in this subject. Now, we are all polite to one another, and we do not throw stones, and otherwise it's okay. But if you look at this group, Dimitar Sasolov is an astronomer, and he has his view of things. Jack Szostak believes in RNA, and John Sutherland believes in basically life comes from soup. And I don't agree with either of them, and they think I'm irrelevant, and we get along fine. But it is an area, unlike much of what you've heard, in which there is no basic coherence to the field at this point. It's completely chaotic. So let me start by posing a problem. And to me, this is one of the most interesting problems in science. And that is, I look at a cell, take your cell, and the cell is composed of molecules, and the molecules are reacting. I know the molecules are not alive. I know that reactions are not alive, but the cell is alive. So what happened? And that's an easy question to pose, but it's actually not an easy question to answer. And what I want to do is address two parts of this problem. One is, how might it have happened? Because I fall in the category of people who think it was not the finger of God, but rather it was some natural process or processes that made this all happen. But there's also a little question of what the it is and how far you extend that idea of life to things that don't look like us or maybe are not biological at all. And I think one of the charms of the area is it enables you, gives you an excuse to think about other meanings for what life might be. And this is a good time to be doing it. So here are some questions. How, when, where did it start? It has a characteristic which I think everyone has to be interested in, which is at the beginning, things were chaotic. The early Earth was a real mess. It was being pounded by meteorites and hot springs and uncertain tectonic plates and things of this sort. And yet out of that, in this came the earliest forms of a cell, which is a pretty uncertain process. It's not clear what that is or what the earliest part of a cell was. Maybe it was a crack in the rock. An interesting question, one that I'm particularly interested in, is life just a name for another set of complicated chemical processes? And the analogy that I use, and I think it actually carries over, and I'll come back to it, is that I think life is a form of flame, which is also a set of organic reactions which is contained in space, which consumes energy, produces products. We'll come back. Are alive and not alive different? I mean, how do we tell when something has become alive? Are we alone? This is one of those universal questions. I think, as you know, if you hold your finger sort of at arm's length and look at an area of that sort, that's in the order, in any direction that you look, of 10 billion galaxies. And each galaxy is 10 billion stars, taking these numbers approximately. And a lot of those have planets, and a lot of the planets are water planets, is what I think everyone is discovering. And I'm not even talking about the dim stars. So if life is at all probable, biological life of some sort, water-based life is everywhere. But we can't think of how that happens. How many kinds are there? Is Beijing alive? That's an interesting question. So just briefly, a few disclaimers of the usual sort. We're not going to be able to run control experiments. We can't go back and start it over again, just not possible. The fossil record for organic chemistry is weak. Now, we just heard a talk in which you can see things that are carbonaceous fragments of something that was there at the beginning. But one of the eerie things about this area is that there was a period at the beginning of Earth when nothing could have survived that was complex organic chemistry. And then, whether it's a few 100 million years or, time sort of fades at that point. 4.2 billion, some people say, 3.5 billion years ago, some other people say, life was there. So very soon, after the planet cooled and settled enough for life to survive, it was there. Now that, for something that we can't figure out how it happened at all, that's really pretty intriguing. There's some things which I'm not going to talk about here, which are actually quite interesting, but we'll pass this by. Tracing dynamics is hard. When you have records that appear in terms of shells or maybe traces of carbon, you have something to look at. But life is intrinsically dynamic. So the fossils that we would be most interested in are the things that happen. And the things that happen leave no record. So that's interesting and intriguing. And to me, it doesn't make any difference. All of this is, in a sense, irrelevant. The question that we can address is, how might life have happened? How might it have happened? We're never going to answer how it happened, but I don't care. Because if I can think of ways in which spontaneously I can envision mixtures of chemicals becoming a primordial example of a cell, I'm fine. That's not going to happen in my lifetime, but someone's. Okay. A short history of organic chemistry. So, organic chemistry started with what we're all familiar with, methane and steroids and whatever, which all have the characteristic they can be simple or complex in structure, but they're trapped metastable states in an oxygenated atmosphere. And I say that simply because thermodynamically we should all burn, but we don't. So we're trapped, we're metastable. There has been a more recent phase which has been focusing on non-covalent chemistry, weak bonds, and molecular self-assembly. And there are a whole bunch of those, and those are quite interesting and relevant, but they're particularly relevant in a fashion I'm going to come to in a moment. And then biology and a number of other areas have the characteristics that they're dynamic, in the sense that there's nothing actually happening that's interesting unless something is happening. It's the happening that's interesting, not a sort of existence. We don't care about silicon, we care about processing information. So these are now complexity of any sort we can rationalize. This is a non-covalent assembly, and it has this characteristic. And you say people with the skill to do this and this surely can work out the question of life. But as my mother, who was a cruel but intelligent woman, would say, you can never change anyone's mind, you just wait for them to die. So for those of you who are young, this is a good sign. And then Pauly puts it, science progresses one funeral at a time. Okay, so we haven't, there are a lot of ideas springing up at this point, but we haven't quite gotten over the hump. Okay. Now, what came before the beginning? And before the beginning, I mean before there were cells, and I don't know when there were cells, so I don't know where before the beginning was, but you get the idea. The idea is that there was a lot of chemistry going on, and I'll show you a little bit about that, but I think that's indisputably true. The molecules coming from space, the molecules being formed in vents and things of this sort, had lots and lots of opportunity to form even relatively complicated molecules. So there are many candidates, and I think you can make an argument that everything that occurs in metabolism now could have formed plausibly from things that were there. And then when something went on, and this is the interesting question, of organization of these into networks or into what I'll call proto-metabolism. And it's this latter question that has so many interesting competing ideologies. And I'll personalize them by my good friends, Professor Sutherland and Professor Shostak. Professor Sutherland's view, and John is a really good organic chemist, is that he can take HCN or H2S or whatever, he can make anything. And so he says, don't worry about the fact that I need skilled postdocs in very carefully regulated conditions. give me an ocean in a few million years and it'll all happen. And out of that will come RNA. Okay? That's one point of view. A second, and this is the Shostak point of view, is don't waste time with all that stuff at the beginning. Just assume RNA and go from there. And there are points of view in both of them. I mean, a good synthetic chemist will do good synthetic chemistry and point out how powerful it is. A good RNA person will work with RNA and point out how flexible it is. But it doesn't necessarily answer the question. So I'm going to be interested in parts that have to do with what's in between the beginning, the crude chemistry at the beginning, and RNA or whatever came before RNA. And the tricks are the ones that I mentioned. We have to find a way of going from simplicity to complexity. We have to think about what this business of self-assembly means. And there are a whole series of things having to do with evolution and memory and expression and catalysis that are key parts of this. And somehow they have to emerge out of this as well. So, very briefly, the problem is here. There are ways of going from simple chemicals and what geo and astrochemistry have demonstrated to relatively complex molecules, not to RNA, not to proteins, not to the very big ones, but to small ones. And starting from DNA and working backwards from RNA, one can build a brilliant series of technical tricks which enable you to do extraordinary stuff, and we've heard a lot of that this morning. And there's always a tendency to look under the light post where the light is brightest. And that's not a criticism, it's just the way it is. And what's missing is this part in here. We don't know how molecules became RNA precursors and the beginnings of a cell. We just don't understand that. And let me show you two slides that sum up what I think is the core problem. You all know that if you take a cup of coffee and you pour milk into it, it mixes spontaneously. You also think about this in terms of if you take the mixed cup of coffee, it does not, in general, go the other way around. Now, that's the second law of thermodynamics. But if you stop for just a moment and you think about that, it's not quite as simple as saying it's the second law of thermodynamics, what's actually going on there. And yeah, we can talk about it, but the issues of entropy of mixing and the potential energy of milk falling some centimeters and that being converted to heat and more states, I mean, it's not a trivial problem. However, relative to the question of we know what a chicken is and how to make chicken soup, but starting from chicken soup, which is the same set of molecules, and making a chicken is a more complicated problem. And that's what happened at the beginning somehow. Now, it took a few million years to get to chickens, but that process got started. And once you have cells, the rest of it, as far as I'm concerned, is okay. I sort of understand how that can happen. I don't know how you get to cells. And I want to emphasize that I'm not going to tell you at the end of this what the answer is, because not only do I not know what the answer is, but I don't know how to think about the answer. Now, what about self-assembly? Because the idea is going to have to be that I have molecules or reactions, and these get together somehow to form the early stages of metabolism. I mean, that seems like, how else could it happen? I'm not sure. So do we know about this with molecules? Yes, we know a lot about self-assembly with molecules. And so what happens with molecules is you have two entities, whatever these might be, and there's a potential function for attraction and a potential function for repulsion, and here's attraction and here's repulsion, and where these cross, you end up with something that looks like that with the right circumstances, and that's self-assembly. And by the way, for this to work satisfactorily, you really have to have that step reversible, but that's a technical detail. So we got that. This is just examples of, this is just examples of things where you have hydrogen bonds that direct what's going on, and the characteristics of the reactions involved at the beginning, not so clear. But the question of what happens when you are involved with reactions is not clear at all. What is self-assembly with a reaction? And I want to show you two pictures, two slides here, to make a point that is important between complicated, this is complicated but not complex, and the next one is complicated but not complex, and life, the formation of life, is complex. So What is this? This is just beads of gas coming out of a bubble generator. And what's sort of interesting is they form these intricate patterns, streams of bubbles, which are dynamic and do what they do. Do I know how that happens? No. Do I know how it happens? Yes, it happens according to the Navier-Stokes equations. It's just I can't solve the Navier-Stokes equations for this. So the basic issue of do I understand it and do I understand it are two things entirely different. One is there is a mathematical description of what goes on. The other is that there is not a deep understanding in an intuitive sense of what goes on. And this, if I can get it to play, is also quite an interesting story. What this is a series of blue and colorless beads. One is Teflon and one is nylon, and I think the nylon ones are dyed blue. And they're simply rattling around on a piece of paper in a shaker. And the reason for the shaker and the beads is not particularly relevant, and the fact it's jerky means that we've intentionally built this system in such a fashion that the motion is random so that we can't synchronize the camera with the motion. But you notice as it's been going on, something's been happening. What's been happening is that it's been ordering spontaneously. And the ordering will continue until you get to, let me just go to the end. You think this is a good idea?
It's magic.
Let me find a way of getting to the end here. Now, it won't do it, so let me go back one. The thing which is interesting to look at, what's interesting to look at is the very first phases. And I say that for the reason that what you're looking at is essentially the early stages in formation of a Coulombic crystal. And we actually don't know anything about these things. We don't know what nucleation looks like. And if you look at this, you can find nuclei forming and falling to pieces. There are no particles in this. This is crystallization. The reason it's happening is that the blue and the white beads charge differently, tribocharging by rattling around on the surface. This is also complicated, but it's not complex. That is, there's no new physics in this. We understand what's going on. But now compare it with this, in which I go from something that looks like that, which is a not implausible place that life might have occurred, to chicken soup to chicken. That's complex. I don't know what happened there. And that's the basic problem. So let me tell you how we're approaching this problem. And the issue here is that from a kind of phenotypic point of view, you can think of life as this. One cell becomes 2, becomes 4, becomes 8, and so on. Got it. That's what cells do. If you look in more detail at the machinery that makes this happen, it's all this stuff that's over here. And even simple organisms have complex metabolism in them. So somehow one has to have gone from relatively simple molecules, which I think you can make from prebiotic precursors, to metabolic cycles. How do you do that? And as I say, the answer is we don't know. By starting with the RNA world, what you're doing is to assume a molecule of great complexity and sophistication, and you're assuming that it can both provide memory and catalysis. The trouble is that RNA is actually not very good at memory, and it's not bad at catalysis, but only for phosphates. So, maybe this doesn't even hold water too well. One of the kinds of things that you can do is to look at simple forms of life and look for hints that there might be something that doesn't require RNA. And there are lots of them, and I'll just show you one because we use it as an example. That is, it's peptide biosynthesis that does not involve a ribosome or anything of that sort. And this involves a protein, which is complicated, but to this protein are attached via thioesters amino acids, and there's a kind of chain mechanism in which this transfers to that, that transfers to that, that transfers to that, that transfers to that. It's a sequential chain elongation, but it's used by many organisms, and it doesn't involve any of the complexity of RNA, DNA, or the ribosome. So you can find many examples of bits of stuff that make complicated molecules but don't require any of the complexity of RNA and DNA. So we happen to be interested in this for reasons that I'll come to in just a moment. But the idea that one has to have the metabolism of a full-grown cell right now is probably not right. And by the way, I point out that if this makes mistakes in whatever is involved in this process, you get something different here, but it remembers it. So there are many of these. This both does catalysis and also does memory. One of the competing ideologies in this area, I've talked about RNA, but there are people who maintain it all started with carbohydrates. There are people who maintain it all started with formaldehyde. There are people who maintain it all started with lipids. And there are people who maintain, in this case the Duve, that it all started with thioesters. And thioesters are particularly good to work with for a reason that I will just sketch here for those of you who are chemists. And that is, I said that self-assembly had to be reversible. And you can get this kind of interchange reaction in which that group exchanges with another in solution to give this relative to its rate of hydrolysis by about 150,000 to 1. So it's a very good reaction for this kind of process. Now, based on that reaction, we ask the following question. Can we design a reaction which has the complexity of a simple metabolic cycle, which doesn't involve anything very complicated? That is, it doesn't involve proteins or enzymes or things of that sort, or is it just completely out of the possible? And what is a simple metabolic cycle? Here's a simple metabolic cycle. This is the Krebs cycle, and it has a thioester in here, which we're going to use. It has something which is an iron-sulfur cluster, which could easily have come from a crack in the rock because there was lots of iron and sulfur in there. It has this kind of molecule, a little trick in wondering where the sulfate comes from, but here's this again. CO2, a lot of this is pretty standard stuff. So it looks easy, but it's not. But let me show you how we think about trying to think about model these kinds of things. And to get in the right frame of mind, I would like you to look at three different sorts of phenomena. We're interested in this. This, of course, is the final answer. And what a cell does is to take nutrients and glucose and maybe oxygen today, but not at the beginning. And it burns the nutrients to make more cell, and it excretes waste in the form of carbon dioxide and water. What's a flame? A flame takes a simple starting material, which is methane and oxygen. You can ignore this kind of stuff for this discussion. Mixes it with oxygen, but the question of whether it's oxygen or something else is probably irrelevant here. And you get a spatially contained structure, which is called a flame, which will propagate. If you have other sources of gas around, it will move into those directions. It produces light and heat as opposed to cells, but it's just a different kind of chemistry. So in a certain way, I am saying that a cell is a highly developed flame in which the chemistry that's going on, the free energy that's being generated and dissipated, is being used in metabolism, not being used in generating heat. Now, this is, I know, not a direct analogy, but it's one that we find useful in thinking about things. So how do we model this? We model it using a trick which the chemical engineers use, which is that we have a small reactor, a continuous stirred tank reactor. We feed it reactants so they don't diffuse in. It does something, it's stirred internally, and then it gets rid of products. And what we're going to look for is a phenomenon which is a complex phenomenon, but which we can follow easily, which is oscillatory behavior. And oscillatory behavior is not easy to find in organic chemistry. So here's what we're modeling, and here's what we have. We have our small reactor and a stirrer and a detector and the rest of this kind of thing. The details are not very important. I'm very proud of this slide because if you are not an organic chemist, you can't make any sense of it at all. If you are an organic chemist, you can't make any sense of it at all either because it's just a bunch of reactions. But there's an interesting point in this, and that is that I'll come to the better way of thinking about this in a moment, but this part of things is an auto-amplifying reaction. That is to say, each time you cycle through that, you start with 1 mole of reactant and you make 2, and then 2 becomes 4, and 4 becomes 8. So in that very casual sense, it has one of the characteristics that I mentioned when I talked about the cell. Now, when a lot of this was worked out by a man named Sergei Semenov, who is now working at the Weizmann Institute, a really, really smart guy, whom you all will meet and appreciate at some point. But Sergei is Russian, and he's also a good organic chemist. So for Sergei to do organic reaction design and to do differential equations, it's all fine. I don't like differential equations that much. So the way the chemical engineers deal with this problem is to use control theory. And that is, rather than looking at all the pieces in terms of the detail, they basically break it up in terms of boxes that have function. And so the functions that we need here are we need an autoamplifier, and I showed you that we had an autoamplifier. You need a trigger for the autoamplification, and then you need something that washes things out. And so what happens is that we feed the reactor a potentially autoamplifying reaction. We feed it at the same time things that set off this autoamplification. The whole thing goes to completion, and then we wash it out. And what happens when you do that is this. And so you have a behavior that's as complex as, but not the same kind of thing as, what you would get from a simple metabolic cycle. That is, you have an oscillation in time in a dynamic system that does what it does. Now, we're going to pass by these parts, which are interesting, but probably not too relevant to the discussion. But if you look at one of the parameters that you can use, which is the space velocity, and then the period of this oscillation, it's pretty delicate. This reaction is pretty delicate. So if the space velocity is too low, you get just an initial oscillation and then nothing. And if it's only about 3 times higher, 2 times higher, you get nothing. And it's only here that you get sustained oscillations. And I'm now going to show you something that's actually quite interesting, which is that this deals, in a sense, with Darwin. And the question is, Darwin was, let's say we interpret Darwin in terms of things like finches in highly evolved organisms. But at the beginning, you had all hell breaking loose. I mean, volcanoes and oceans and meteors and stuff like that. And the question was, what was fittest? And my guess is that fittest was not specialized beaks that allowed you to pull seeds out of trees, but rather the ability to withstand extreme variations in reaction conditions. And so what we're interested in doing is look, rather than at fitness in terms of optimization, we're looking at fitness in terms of robustness against variations. And go back to this particular system, and what we're going to do now is just to try a couple of simple experiments. One is to take the system that I showed you before, but we're going to work with multiple thioesters. We're going to make a messy starting mixture rather than a clean one, because I said it was pretty delicate. And when you do that, the result was actually, to me, unexpected. And you can summarize the results down here in terms of this truth table. If I work with glycine, if I work with alanine ethyl ester, this compound, I find that if I have nothing present except this, it oscillates. That's the reaction that I showed you. If I work with glycine, which is just missing one methyl group, it doesn't oscillate. But the interesting thing is that if I mix these two, it continues to oscillate. So that by making the mixture a mixture of something that doesn't oscillate on its own and something that does oscillate on its own, I continue to oscillate, so it's robust to that kind of change. More interestingly, if I work with another set of things, and I have glycine, and I have this material here, which is not correctly drawn there, but it's aminobutyric acid. If I look at glycine just by itself, as in the last slide, it doesn't oscillate. If I look at aminobutyric acid, it doesn't oscillate. But if I take these two things, neither of which oscillate, and mix them, they oscillate. So that what has happened then is that you get complex behavior out of a mixture of things in which you don't see any behavior that's complex at the beginning. So it's not only more robust if it's a mixture, but it's also, there's emergent behavior. This is not what we expected. So this is a poor imitation of a metabolic cycle, but it's the same degree of complexity, except it misses the enzymes. So I think it's an interesting start. Okay, let me move to the last section. That is, I've said that I think that one of the ideas that's beginning to emerge is that mixtures may make things more robust than highly evolved systems. And if you look at all of this kind of thing that we're doing here, one of the interesting questions is, how do we know whether we're making progress toward an answer here? I mean, how do you measure relevance in a system of this sort? To what extent does this look like that? And I don't think the answer to that is very clear. But when you begin to ask the question of how do you know when we've succeeded the origin of life or metabolic cycle or whatever, there are a couple of things to think about. One is that Darwinism requires error-prone replication, basically. It requires mistakes that allow the formation of new species. And so it requires memory and information and error. And you also need emergence. You need new forms of behavior. And that leads us then to a series of questions which are now non-scientific but interesting to think about in this context and others. The first one is, if we're going to talk about the origin of life, we might ask, what is life? The problem with that is that, let me tell you, I talk to my friends who are physicists. I have friends who are physicists. And I say to them, since I'm a chemist and chemists work with nuclei and electrons, I say, what is an electron? And they say, that's not a legitimate question. The question that you can ask is not what is an electron, but rather what it does. And you can ask the same kind of question with life, maybe. Not what is life, but what does life do? And if you do that, you broaden the definition in a pretty interesting way. A lot of things in principle could become alive, which are not alive before. Emergence. Now, emergence is one of those words that arouses all kinds of peculiar emotions in people, particularly physicists again. And, you know, the question is emergence something that we just don't know what it is? You know, we haven't seen it before, but it's same old chemistry and physics. Or is there actually something new there, something we haven't thought about? And I think most chemists and physicists would say that this is a problem in which there's probably nothing fundamentally new there. But having said that, do we understand the behavior of complex networks? And to me, this is a little bit like the question of statistical mechanics. If we could understand molecules, would we understand statistical mechanics? No. If we understand statistical mechanics, do we understand thermodynamics? No, not really. So the question of asking whether there's something in these nonlinear networks that is going to require us to rethink something important, I don't know the answer to that, but I think it's a fair question. So does it make a difference? Yes, because if we only have to look at those ideas that we already have, we don't need to worry about new stuff. But this question, I think, is really quite an interesting one. And that is, if we understand something in principle, as, for example, my stream of bubbles, which is just a Navier-Stokes equation being worked out, but we know we don't understand it. Or we understand, we see the Schrödinger equation, which we all solve every day. But do you really know what's going on in quantum mechanics? No. Same thing in special relativity. The thing is that we're moving into that phase in science in which mathematics is beginning to exceed to some extent what one can easily understand. And whether there's some of the same kind of thing here, I don't know. And then my last question, which I think is simply a point of this, does the Schrödinger equation predict true love? Well, I don't think so. Not that I didn't do that calculation. But, I mean, true love is a product of matter, and matter follows quantum mechanics, so that if you're a true reductionist, then true love is just an expression of the Schrödinger equation. And it leads you into interesting problems in what is knowable and what's not knowable in reductionist science. This is, I don't think it's a trivial problem. Then one last subject. That's an issue that I think is really, really quite interesting and quite relevant now. And that is, can we tell the difference between alive, not alive, dead, partially dead, other than alive? I mean, are these all the same thing, just different flavors of something? Are they actually different? And here's some examples. Is a frozen embryo alive? Is a megacity alive? Is the World Wide Web without people alive? Is the World Wide Web with people alive? Are the 2080 Internet of Things, a virus inside a cell, a senescent cell, an ex-nucleated cell, the most AI of 2080? The issue is that these things all have the characteristics that they will undoubtedly be able to do interesting things, some of them that we can't do. I don't know if that makes them alive or not. I mean, I know a lot of people who are not by any standard that I can use alive. So that question of where I draw the line between something. But a city is interesting because it's, you know, I am a contained object that into which the mass transport brings food and oxygen, water. There's waste. There's information processing, various and sundry repair things. Sounds exactly like a city to me, except you have people rather than cells. So could there be life that is completely different from ours? And you know, the first question, the one that a lot of exobiology focuses on, is how many planets are there that might support our kind of life? I think the answer is a lot. Martin Rees has an answer that we may never see any of them, but that's a different issue. The question of what are we doing? We're looking at the question of how life might have happened, but I don't know about that. And then someone raised the issue of Copernican revolutions. And if you think about that problem, you know, the first Copernican revolution made the fact that Earth is not the center of the universe. We got it. I think we're dealing with the second Copernican revolution right now, which is that I don't know what's going to emerge from very large computer networks and what happens, but it's certainly going to be able to pass the Turing test. And is it alive? I couldn't tell you, but it may be sentient but not alive. And then the third one is there life everywhere? If there is, then, you know, we're not the center of the universe, we're not the most intelligent thing, and we're actually common as grass, and it actually puts us in a somewhat different situation in terms of overall what's going on. So these are all good and interesting questions. Now, just a couple of things because they're relevant. I think I have mostly thought about life as something in which there's a long stage in which maybe life was not very complicated, and then it got to be complicated, and we're somewhere up in here. But if you look at the sort of plausible mathematical formalisms for that, you could say that you start out with something we're doing here, and it would be not alive, and all of a sudden a switch would throw and it would be alive. That would be binary. Or you could have it exponential and continuing, or you could have it be something that was, I don't know, sigmoidal. You can have bifurcations of various sorts, this and this. Maybe it becomes more alive, but then there's a switch that throws and it moves to here, and it becomes less alive as it moves back down there. I mean, speaking as someone who's almost 80, this has a certain, I understand the process. You become more alive, and then at some point you're really quite lively, and then, you know, I don't want to go into it. It's too depressing. Then there's, you know, there's a period where you're alive. There are lots of different things of this sort. And, you know, mathematicians have many ways of thinking about functions that have different characteristics. How many of those could be applied to different forms of life? It's a neat question, to which we can't answer the question. So I think 2 slides to finish. Since we're interested in the basic question of what's needed and what the opportunities are, we need to reintroduce understanding deeply about thermodynamics, particularly entropy and information. I maintain, maybe incorrectly, that the key element to life from a chemical point of view is catalysis, because catalysis is the ingredient that enables reactions to talk to one another and is the key element in self-assembly of reaction networks. Statistics and probability, if any of you don't know what the Leventhal paradox is, come and I'll tell you later. Combinatorial chemistry and things of that sort. How many different reactions do you have to run to come to a new reaction of a given degree of improbability? I don't think we have any idea about that. Networks, Darwinian ideas, I think Darwin was one of the greatest of all scientists, but how do you apply his ideas in new directions? And then, new mathematics. How do we think about what the new mathematics should be? And how do you get people who are organic chemists who have normally chosen organic chemistry because it's as far away from mathematics as they can possibly get? How do you put them together? And then missing science, we don't have a plausible mechanism for self-assembling of networks, and we don't actually really know how networks work. It's a real blank. Dissipative systems are a real blank in chemistry. Sources of energy. And we have lots of sources of energy, but if you look at the way in which you could take, for example, a thermal gradient and harvest that free energy difference to do something, we actually are very short on having good ideas about how to do the harnessing of gradients. Enzymes, other catalysts, sets of reactions that work under common conditions. The amazing thing about me is that all my reactions run at the same temperature in water. And that's not the way organic chemistry goes. And that's made possible by catalysis in large part, but we don't understand those systems. Concepts for the origin of evolution in memory, error proneness, the rest of these kinds of things, Darwinianism we've talked about. And then a sense for the coevolution of nucleic acids and proteins from plausible precursors. How do you build macromolecules of any sort plausibly? It's not as straightforward as it seems to take a nucleic acid precursor or a protein precursor and just dehydrate it. You don't get high molecular weight stuff generally. So that's the story, and I leave you with this. These are all the people who did the work. But the basic question is, how do you take a series of reactions and a series of molecules which are not alive, and then something happens? How does that happen? And we don't know. Thank you very much.
Questions, comments? I would like to begin. Professor Sternberg. Professor Sternberg drew our attention that atoms and molecules as we know them constitute less than 5% of the universe. There could be life that we don't even begin to have the words how to begin to think about it.
Think about what E. coli thinks about, for example, the Schrödinger equation. And E. coli doesn't spend a lot of time imagining the Schrödinger equation. So the idea that we have a complete set of, why not? I can't know what I don't know. So I completely agree with you. So is there forms of life, dark matter that's alive? Why not? Maybe they may be sitting here chuckling at this entire performance.
Listening.
Yeah.
Yes, please.
George, we've talked about this problem, and we're obviously not going to solve it here. But I wanted to just raise the issue of comparing the flame and your bacterium. Mm-hmm. One big difference between the flame and the bacterium is that when you look at the bacterium, wherever you look in there, you see function. Function is everywhere. There's no function in the flame. Now, the question is, where did function come from? Jacques Monod said this is the central problem of biology, to explain how function and purpose emerge from an objective universe with no function or purpose. So it seems to me that going into the lab and doing lots and lots of experiments isn't going to solve that kind of problem. We have a deep theoretical problem here that somehow physics and chemistry has still not been able to give us an understanding of these concepts, a concept like function, which in principle does not exist in physics and chemistry. Atoms and molecules don't have functions, and processes don't have functions. So
But
what's
remember,
your response to we need something is missing in the theory?
well, that's the question of, is there something here we don't know? Or is it just by random chance reactions got together, formed networks, the networks found a way of improving their performance by being autocatalytic or something of that sort? The autocatalysis went in the cracks in rocks where the walls were just naturally lined with catalytic species. I mean, I don't know how to do it. But remember, if you tried to cook a steak over a pot of bacteria for all their function, they wouldn't do it. So we're providing the idea, the concept of function, both in the case of flame and in the case of bacteria. I mean, I know you don't agree, but that's all right. I mean, you're exactly right in what you say, and that's the key of the problem. We don't know how that happened.
I'd like to ask a couple of questions. One, it seems to me that if you have linear theories, like the ones we accept as being the foundation of physics, quantum mechanics, general relativity, then it seems to me there is no emergence, because with linear theories, the whole is simply equal to the sum of the parts. So therefore, you need nonlinear equations, nonlinear mechanics, chaos theory, and so on. And so it seems to me that that's why ideas like catalysis are important, because of the feedback. You need something nonlinear for patterns, for something to emerge, which is not equal to the sum of the parts. Seems to me the first thing. And the other thing of the concern about
I'll tell you what, we're going to do this in the following way. I'm going to answer this because the thing to remember is that if you're talking to a speaker over 65, you don't ask two questions and expect the first one to be remembered. So, I mean, you're absolutely right. But it leads to the question of, if I look at systems of coupled nonlinear functions, if I look at that kind of system, and I agree completely, as you know, I look at a molecule and I don't end up with statistical mechanics until I've done the statistical mechanics. Are there new things there? I think there are new things in the behavior of networks that are complex and involve many, many moving parts. So I think there is something actually quite new, but it may not require coming up with quantum mechanics.
Well, the other thing which is related to this is the second law. And it seems to me that it's got nothing to do with this really, because what we're talking about is steady state, not equilibrium. And these are different things. You can have patterns which are very far from equilibrium. A flame may not be at equilibrium, but it has a pattern. A swirl in a river is not equilibrium, it's a steady state phenomenon. And so patterns emerge from steady state and are not in contradiction with the second law.
I didn't say any of this was in the second law, was in contradiction, because we know that it starts with the sun at 10,000 and it ends up with photons scattered around the universe at 4 Kelvin. So, I mean, it all just involves a downhill cascade. It's a question of what is involved in terms of providing subcycles in this, which you can call functional, you can call complicating, or you can call complexity, whatever it is. But what's not clear is how that happened. How did it happen spontaneously? And that, I think, is the key problem in the origin. And the answer is, I don't know, maybe you do.
We thank George.
Sunney? No, Sunney gets to ask a question.
In the 18th century, Leonardo da Vinci asked a question, what is water?
What is water?
More than 500 years, and we are still a little bit hazy about it.
No, some of us are very hazy about it.
Is there an analogy?
Well, you know, of course there is. But, you know, asking what is life, we may be able to answer the question, but the question is, where did it come from? That's a harder question. So, yeah. Sunney, you got a question?
George, cells have functions, but they also replicate. So in all the studies that appeared, has anybody succeeded in making a self-replicating system, not using DNA, RNA? I mean, if there's an example, then there's a possibility, another possibility.
If you take, I mean, we're interested in flames, and one of the interesting things that you can see with flames is that in old gas burners, as you turn down the gas burner, you will reach a point where you have a circulating flame going around the edge. That's self-replicating. And it is something in which what happens is exactly what I showed you in that auto-amplifying system. You have a series of flamelets. Each flamelet burns out its fuel, oxygen mixture, and it can't replenish until some time has gone past. So there's a progression through a series of things, but the progression is continuous. And if you've ever filled your house full of methane and then light a flame somewhere before the house collapses on top of you in the resulting explosion, you do remember that that's a propagating flame. Right? Yes, you look reluctant to think about that, but that's okay. All right.